Well, hello and welcome to Online Lecture 7B here in your Biology 112 semester. Guys, today we're going to be in Chapter 35, looking at slides 11 through 40, basically looking at the general understandings of what we know about flowering plants, just basic fundamental stuff here, guys, in Chapter 35, all looking at angiosperms, looking at how they grow, how they develop, how their body is laid out, etc. We'll also look at a video on the flowering plant life cycle, which we'll get to shortly. Let's go ahead without further delay. Let's get into slide 11 here, guys, and begin our study here, and actually kind of continuing our study of what you'd started in Chapter 35 in class. So this is just showing you an overview of the body plan of a flowering plant. And if you look kind of carefully, you can see, at the top we have our flowers, which is actually considered an organ system. Does anybody remember why a flower is considered an entire organ system? One flower? Yeah, because you've got lots of organs, right? Sepals, petals, stamens, pistils, at least or up to 4 floral organs there. Buds are actually considered organ systems because usually you have your undeveloped shoot, along with some maybe immature leaves, some bud scales. There's several different organs all bunched together in that little bud. Think of a bud as a immature shoot system. A branch is considered an organ system, right? That's not quite as hard to see because you could see clearly a leaf is considered an organ, the stem is an organ, so that's already 2, plus your flowers, which are multiple organs bunched together in another organ system. So the whole branch itself, of course, would be an organ system. And you can, of course, see the leaf is just an organ, and a stem is just an organ. Leaf is just a bunch of tissues working together for a common purpose. And if we zoom in on the leaf, you can see the tissue here. And the tissue, of course, is made up of cells. Right, guys? So cells make up tissues. Tissues make up organs. Organs make up organ systems. Just a review from good old Biology 111. And you can see everything from here on up is part of the shoot system. And, of course, the root system is down below. The root is considered just an organ, though. So you'll see some roots can be modified into doing other tasks like storing very large amounts of starch, like in the case of some of the very thick tap roots. Some root systems function even in gas exchange, but your typical root is there to absorb water, minerals, and to anchor the plant, maybe to store some extra sugars, but that's just the root system, just consisting of one organ type. And of course, within the tissues of many plants, you find cells and specialized cells as well. So that's just like an overview of the plant body. So flowering plant body, to be specific. Here are just some examples of some flowering plants. So you can see brachychiton. Basically, all your trees are pretty much flowering plants, guys, except for your pines, right? The various conifers, your gymnosperms, which we had studied before. But this is definitely an angiosperm. You can see it's got, all the basic floral parts we had talked about. Don't really see the flowers coming out quite yet, but it does have all the parts of the plant that we had just previously looked at on slide 11. And you can kind of see that there. And it's a rather large tree. And then we have from that all the way down to little lemna, which is called duckweed. You guys have probably seen this stuff floating around on the pond surface. And this is one of the smallest of all the flowering plants, where you could see it's got leaves, it's got very tiny little simple roots, got all the basic parts, very small stems. So there's quite a bit of variation in form and quite a bit of variation in general body design in these flowering plants. But they have those basic parts that we had looked at in the previous slide. So let's now move on to slide 13, and let's begin to look at some of the distinguishing characteristics between monocots and dicots. So when we look at flowering plants, realize we can take all flowering plants and divide them into 2 big categories. Now, there is a third group called the magnoliids, but the most common of the flowering plants are either going to be dicots or monocots. And remember, those names are just short for monocotyledon and eudicotyledon. So if you're a dicotyledon, that means you have 2 cotyledons, which are seed leaves. So there's 1, there's 2. In the dicot, there's only 1 in the monocot. Flower parts, remember, and the dicots, they're usually multiples of 4 or 5. And so on this particular flower, we have 1, 2, 3, 4, 5. So definitely a dicot. And then this flower has 1, 2, 3 petals. That's a multiple of 3. That's typical of monocots. So monocots, multiples of 3, dicots, 4s and 5s. How about if you were to do a cross-section of the stem? Well, you would find that the vascular bundles, or, well, it sounds kind of fancy. How about we just call them veins? The veins are arranged in a ring in the stem of a dicot, but in monocots they're very scattered. Generally speaking, guys, monocots are not quite as complex or organized, so don't be surprised that you'll see things not as organized. Like on the monocot, you'll find vascular bundles scattered all over the place, whereas in the dicot they're arranged in a nice, neat ring. Okay? Also, it doesn't mention it here, but the pollen grains in monocots usually have 1 pore, and dicots, I should say, they have 3 pores, 1, 2, 3 pores in the pollen grains of dicots, only 1 pore in the pollen grains of monocots. Right, so also, I guess, oh, they are going to show that here. Okay, so next slide, you can see that even better. So you have 3 pores or slits here in the dicot, 1 pore over here in the monocot. Of course, the root system on dicots is a taproot, very thick main root with lateral roots kind of coming off, whereas in the monocot, there's not one main root, lots of thin, fibrous roots, and they're what we call adventitious. Does anybody know what that means? Adventitious? Okay, adventitious means that the root comes directly off the stem or even a leaf in some cases. All right, so that's a little different. So these adventitious roots actually come right off of plant organs, again, such as stems or leaves. And, of course, they're all very thin. You can't see that one main root there. Venation patterns, of course, with the dicots, they're very netted or branched, as you can see here. And then in monocots, they're typically parallel. So most of your grasses, of course, guys, would be monocots. So that's one way you can remember the differences. But most flowering plants out there are dicots. All those deciduous trees, they're all dicots with few exceptions. Okay? All right, guys, well, let's go ahead and get into the next slide, slide 15. So let's really get into how plants grow and develop. We've looked at their body plans. We've looked at some of the differences between 2 major groups here. Now let's get into how they actually do this amazing thing of growth and development. I'm sure you've been very impressed with the ability of plants to grow. They can grow very fast. And they can also grow very, very tall, very heavy as well. So how do they actually do that? It's amazing. First, we have what's called vegetative growth. This increases the size of the shoot and root systems. Notice in vegetative growth there's no mention of flowers or fruits. That's what we call reproductive growth. So vegetative growth is all the growth of everything in the plant body except for the reproductive parts. Again, the flowers, the fruits. So in vegetative growth, it starts at buds, especially in the shoot system. In the root system, we don't have buds, but in the main part of the plant, at least the part that you see, it all begins in buds. A bud is actually a immature shoot system with a miniature shoot and dormant shoot apical meristem. And remember, the apical meristem is where what happens? Anybody remember? Yeah, the apical meristem is where there's a whole lot of cell division. Lots of cells dividing, lots of undifferentiated cells, cells that are not specialized. And then those cells will eventually become specialized through the process of development. Under favorable conditions, the buds produce new stems and leaves. However, there are some buds that will turn into or develop into flowers. But most of the buds you see on a plant are going to develop into new stems and leaves, which is the shoot system. Now remember, buds are an organ system because they contain more than one organ. They will have an immature stem and leaves inside of them, along with bud scales on the outside. The bud scales are mostly there in plants that live in temperate areas, places that can get cold in the wintertime, to protect the delicate immature organs underneath. In tropical areas, you don't see the bud scales so much. And what we see in vegetative growth is something called indeterminate growth. So when it comes to vegetative growth, this is the type of growth we see, indeterminate, which means you have a continual production of new stem tissue and leaves as long as conditions remain favorable. So you can get really huge organs. You can get very large stems, very thick stems, stems that branch because we have this indeterminate growth. Now in individual leaves, we don't have indeterminate growth. A leaf is programmed to get to a certain size and stop, whereas a stem will grow and continue to get bigger and bigger and bigger and bigger. So stems exhibit indeterminate growth as well as roots. However, flowers and fruits and leaves have determinate growth. They grow to a certain point and just stop. They're genetically programmed not to get any bigger. You see? All right, so let's now look at reproductive development. So we looked at vegetative growth, things like stems, roots, leaves, etc. Now let's look at the development of those reproductive structures, flowers, seeds, and fruits. Well, flowers and floral buds, so again, we do have buds that turn into flowers, not just shoot systems in general, like stem and leaves. These are reproductive shoots that develop when the SAM, or shoot apical meristem, produces flower parts instead of new stems and leaves. And so this would happen typically in an annual. It will happen towards the end of the growing season. In a biennial, you'll see a lot of reproductive growth in the second year of life. In the first year, they put most of their energy into vegetative growth, and then their second year, they'll go into reproductive mode and then die. Now, the perennials, which live 3 or more years, will produce reproductive shoots through floral buds, usually every single year, typically in the spring and the summer, although there are exceptions. And of course, conditions have to be favorable for that. The plant has to be healthy. Okay, guys, let's now go on and again look at some examples of determinate growth. So flowers are produced by determinate growth, growth of limited duration, and a growth of a limited size. So we get to a certain size and we simply stop growing. If flowers had indeterminate growth, flowers would be enormous. Flower tissues enclose and protect tiny male and female gametophytes. Remember, your tiny male gametophyte would be pollen grains, right, guys? And what would be your female gametophyte? That would be a tissue found within the ovules, within the ovary. So that's where you'll find your gametophytes. Again, male gametophytes along the anther, where you have your pollen grains, which is basically immature male gametophytes, and then your female gametophytes, which again are found down the base of the ovary inside of the ovules. And then the fruits enclose seeds and function in seed dispersal. So the whole purpose of fruits is to encourage seed dispersal. And I think all of you know by now that fruits come from flowers. Once your flower has been pollinated and the sperm come out of those pollen grains and fertilize those ovules, the ovules develop into seeds. And then the ovary develops into your fruit, right? You guys remember some of the stuff. A lot of this, I'm sure, is review. So let's now go on to some of the different types of plants. I mentioned this just a second ago. We have annuals, which grow and die all in 1 year. Typically, annuals spring up in the springtime. They grow rapidly throughout the summer, and then they die in the fall. But right before they die in the fall, they're going to produce flowers and, of course, small fruits and seeds, and then die off. Biennials, examples of annuals would be basically most of your weeds. Now, biennials, again, they don't reproduce their first year. They put most of their energy into vegetative growth, growing their leaves, stems, and roots, etc. And then the second year, they'll produce flowers and fruits, perhaps. And then they die off. And then lastly, we have the perennials, plants that live for more than 2 years, 3 or more years, typically producing seeds each and every year. So you will see the perennials will be basically your trees, guys, your shrubs. And there are other examples of that, but mostly these are your larger flowering plants. So we have annuals, biennials, and of course the perennials, which pretty much summarize all that you see in the flowering plant world. Now, we'll move on now to further discussions of plant growth and development. But let's now zoom in. We've looked at the big picture. Now let's zoom in to the cellular level and begin to examine plant growth and development. So to grow and to develop, you have to have cell division, and growth, of course. Cell division leads to growth, and of course cells also increase in size. Then we'll see those cells become specialized, and then other times we'll see cells kill themselves in the process of apoptosis or cell suicide. So these four processes are going to essentially cause a plant to grow and develop as it should. It sounds strange that we'd actually have cells killing themselves in this growth and development process, but some cells need to go away to allow more mature tissues to develop. For example, a tadpole becoming a frog. The cells within the tail of the tadpole must actually commit suicide in order for the tadpole to develop into a frog. So the same kind of thing can happen in plants. So we also have some additional principles when it comes to plant growth. Development and maintenance of a characteristic architecture throughout life. So we have the development and maintenance. So we're going to maintain a certain body form all throughout life. That takes a lot of effort, takes a lot of energy to maintain that body form. Think about those trees around your house. They maintain a basic architecture throughout life. Also, you may have not noticed this, but they increase in length by the activity of apical meristems, which are only found at the tips. So you can only grow lengthwise, you can only get longer and taller by the activity of apical meristems found at the tips of shoots and the tips of roots. The thickening happens all throughout from the trunk on up, but when it comes to increasing in length, which is called primary growth, that only happens at the shoot tips and root tips, better known as the apical meristems. Now, there's always also a maintenance of population of youthful stem cells in the meristems. So we will always find in a plant at the tips, in those apical meristems, the root and shoot apical meristems, a population of young cells that are undifferentiated that will eventually become specialized over time. And that will always be present in your plants as long as they're healthy. All right? That's very different from an animal. We have youthful cells when we're young, and then we basically sort of maintain through mitosis, replace worn out and damaged cells, etc. But plants continually grow throughout their lives because they always have these youthful, undifferentiated cells, unspecialized cells found in the meristems. And of course, we actually have expansion of cells like we also see in humans, but it's in very controlled directions in plants, and water intake plays a big role. So we'll talk more about how that happens shortly. So these are the four basic activities that will result in the maintenance of a plant's architecture and also help a plant to grow. All right, so let's look a little bit more at the architecture of a plant. You could see in all flowering plants, you have apical basal polarity. What does that mean? That means there's an upper apical pole and a lower basal pole. There's a top and a bottom. This all starts very early in embryonic development. They did some studies and they looked at GNOM or NOM mutants, and they found they became cone-shaped because they lacked apical basal polarity. So just a few genes helped to maintain the apical basal polarity so that when they mutated the GNOM genes, it caused the base little embryos to develop into cone or round-shaped plants, which is, of course, not typical. Apical basal polarity is a good design because it helps a plant get to be nice and tall so they can compete with neighboring plants for sunlight. That's probably why they have that architecture, guys. And then radial symmetry is really common in flowering plants. You'll find a cylindrical shape like in your stems. If you were to cut a stem, do a cross-section, you'll find a generally round shape. You even see that with flowers. You also see that with fruits. There's generally a cylindrical shape, especially if you do a cross-section. Leaves and flower parts are typically also arranged in whorls or spirals. So your petals are arranged in spirals, which results in a cylindrical shape. So this is your distinctive architecture found all throughout the entire flowering plant world. And here are some examples of exactly what we're talking about here, guys. You can see a normal seedling has the shoot, apical, meristem at the top, root at the bottom. You can see there's a distinct top and there's a distinct bottom. And then if we have mutants that have mutations in the genes that code for apical basal polarity, you have this unusual round or cone-shaped sort of structure, which again would not be good for a plant, would not be able to compete with neighbors very well. And here is the cylindrical theme that we find in the flowering plant world. So we have, these are baby leaves here, guys, leaf primordia, and this is the shoot apical meristem here. And you can see, we're starting to see that round shape, right? We have a clear circumference and a diameter as well. Okay, so there are some general themes there. Let's now get a little bit more into the meristems. Now, we mentioned them several times. Well, let's really focus in on them in a little bit more detail. So, plant embryos grow in seedlings by adding new cells from only two growth points, your shoot apical meristem and root apical meristem. If those are damaged, guys, you actually won't grow any taller or longer. You absolutely have to have those in order to grow longer or taller. So all primary growth happens in the shoot and root apical meristems. Now, they give rise to many apical meristems located in buds and shoots and the tips of roots. So we'll start out in the embryo with only one shoot apical meristem and one root apical meristem, but then they will give rise to many apical meristems, right? Like in a tree, you might have hundreds or thousands of apical meristems located in the tips of all those little twigs. And then down in all the branches of the roots, you'll find lots of root apical meristems. So this thing multiplies, and this allows a tree to get really big in both the root and the shoot system. Don't forget that root system can be just as big as the shoot system. So when you look at a tree and you're seeing the trunk on up, there's almost just as much mass from the trunk down below to the tips of the roots deep within that soil. So let's now examine this a little bit more. So as the shoot apical meristem grows, it leaves behind primary meristems as part of primary growth. So the first thing that the shoot apical meristem does is, of course, it's dividing. Lots of cells are dividing repeatedly through rounds and rounds and rounds of mitosis. And then those cells begin to specialize. They come out very undifferentiated, very general, and they start to become specialized, and so they turn into three primary tissues, which we'll get into in just a moment. And that's all part of what we call primary growth, which again is growing in length. Secondary growth is an increase in thickness or girth, and that comes from secondary or lateral meristems, which we'll get more into for test 8. And that most of this information is found at the very end of chapter 35. So we won't get into secondary growth too much, but just know secondary growth is increasing in thickness, and that comes from the lateral or secondary meristems found just inside the stem. Just below the bark, in fact, guys, you'll find all of your lateral meristems just below the outer bark. And it's found actually between the outer bark and the inner bark, which we'll discuss that more. Again, not quite so for this particular test. Okay, so now let's look at the primary meristem. So what comes from the shoot apical meristem and the root apical meristems? We have three primary tissues. The protoderm, which generates dermal tissue, guys. In other words, the protoderm will eventually turn into your epidermis. So the shoot apical meristem cells, many of them will differentiate, start turning into protoderm, and then those protoderm cells will eventually turn into your epidermis, your dermal tissue, your outer tissue, kind of like skin. And then you have the procambium. These cells eventually develop into your vascular tissues. And what are vascular tissues? Does anybody remember? Yes, your xylem and your phloem, which are conducting tissues and also function and support. So xylem carries water and minerals from the roots up to the leaves, and phloem carries sugars, dissolved sugars from the stems down into the roots, from the leaves down to the roots. So those are your vascular tissues. That's all procambium that develops into those. And then lastly, you have your ground tissue or your ground meristem, which produces ground tissues. What are ground tissues? Well, this would be the filler material. So between your epidermis and your vascular tissues, all that stuff in between is your ground meristem. Things like pith, they typically are thin-walled cells. They oftentimes store starch. So, you know, most of that sort of that fibrous area between your vascular tissues and your epidermis, or your outer sort of area of your bark, that's all ground meristem. Again, think of it as filler tissues. So plants' cell specialization and tissue development does not depend much on the lineage of a cell or tissue. So as the cells go from shoot apical meristem, root apical meristem, these undifferentiated cells start developing into protoderm, procambium, ground meristem, eventually developing into epidermis or dermal tissue, vascular tissues, and ground tissues. The whole process of that doesn't depend so much on their lineage. Like, in other words, you know, since I'm a protoderm cell and my parents, if you will, were protoderm cells, I am destined to always be a protoderm cell. It's not so much on your lineage, you know. So if you're a procambium cell, it's not necessarily because your mother cell, your parent cells, if you will, were also procambium cells. It's much more dependent on things like hormones, chemical influences. So what's going to determine whether a cell becomes protoderm, procambium, or ground meristem? Hormones, guys. Mainly hormones. Expressions sometimes of genes resulting in certain proteins. So it's certain hormones, certain proteins that will cause a cell to start to become specialized, to start to become a procambium cell, or eventually a xylem cell. It's not so much on your history, okay? It's kind of like, you know, it's the equivalent of someone's career choice. Is your career choice dependent on what your parents did? Like, are you going to become an insurance salesman because your dad was an insurance salesman? Right? No, not so is the case. In the old days, if your dad was a blacksmith, you became a blacksmith, pretty much. And that's how, you know, basically that's how society operated. But now it has much more to do with your personal likes and your own experiences in life. And so that's kind of how it is with these cells. It's not so much, you know, well, my parent cell was a procambium cell, so I'm destined to become one. No, it's the chemical influences that have much more to do with what cells actually wind up becoming. Okay? So let's now zoom in this a little bit more. So primary stem structure and development. Let's look at the development and the actual structures of these, of primary growth and lengthening. So new primary stem tissue rises by cell division, right? So cells are dividing in the apical meristems and the primary meristems near the bases of the SAMs. So you'll find all your primary meristems, your protoderm, your procambium, your ground meristem, all that is located just downstream from the shoot apical meristem, which is at the very tip of the stem. The epidermis will develop at the stem surface, and it will produce a waxy cuticle, which of course you all know by now it reduces water loss. Plants that live in arid regions develop a thick waxy cuticle. If plants live in a wetter area like here, they develop a thinner waxy cuticle. So the epidermis is right there at the stem surface. And you'll even see specialized epidermal cells like guard cells develop, and stomata. Now, then we have the cortex. The cortex is composed of parenchyma tissue. The cortex, guys, is ground meristem tissue that's now more mature. So the protoderm leads to the epidermis. The ground meristem develops into the cortex, or at least that's one of the things the ground meristem develops into, the cortex, which is parenchyma tissue. It's very thin-walled cells. In parenchyma tissue, it's softer. The cells are thinner-walled, and they're very good at storing things such as starch. So we see a lot of soft, nutritious cells, if you will, in the cortex. And typically, it's composed of only one cell type, just parenchyma cells. So parenchyma tissue is composed of parenchyma cells. And so it's a simple tissue. Remember, simple tissues consist of what? Oh, simple tissues consist of one or only two cell types. And of course, they store starch and plastids. Again, very thin-walled cells, very soft tissues. Stem parenchyma also has the ability to undergo cell division. It has meristematic capacity to heal wounds. So parenchyma cells can easily be manipulated by chemical influences to become totally different cells. So we could actually see a parenchyma cell turn into a xylem cell or a phloem cell. And so we'll see parenchyma tissue is very easily healed. If you damage it, it can heal easily. It can even turn into something totally different, which allows you to graft. Like, for example, guys, I can take a stem from an apple tree and I can graft it into a peach tree, and we'll have a peach tree producing peaches, but then on that one stem, we could actually have an apple form. So you can have one tree produce both apples and peaches. How interesting. All because of parenchyma cells, which are very undifferentiated or have the ability to differentiate into different cells. Allows you to do all kinds of neat grafting. I actually owned a tree that could produce apples, peaches, plums, something else. Gosh, it was really, really cool. All because of the grafting. And you could even take a stem and stick it in the ground. It could become a whole new tree, all because of these parenchyma cells being able to differentiate. So very neat. All right, guys, well, let's now go into some of the other tissues. We looked at parenchyma. How about parenchyma? Parenchyma tissue. Well, this is composed of parenchyma cells. Parenchyma cells are very flexible. You know when you bite into celery and you get like the little veins in the celery and it's kind of chewy? And sometimes they'll even make you gag a little bit, like if you don't chew up that little string, that stringy vein. That's parenchyma. Parenchyma is flexible, chewy. These cells are unevenly thickened. So their cell walls are really thick in some spots, very thin in others. This makes for the nice, flexible, and sort of chewy, if you will, tissue. Sclerenchyma is more rigid. It consists of fibers and sclerids, which are very hard and rigid, so sclerenchyma is much tougher. Sclerids, you know when you eat a pear and you bite into the pear and you get that gritty texture? Folks, that's what sclerids are. And then fibers, of course, make things very tough and strong and almost unpalatable. If you have too much sclerenchyma tissue, you can't even attempt to eat that. We can eat celery because it's mostly parenchyma and just some parenchyma along those veins that are chewy, and it, of course, becomes stringy and kind of make you gag sometimes. I've done that before. Then we have primary vascular tissue. This develops from new cells produced by the shoot apical meristome. So we finally get our primary vascular tissue, our xylem and our phloem. And remember, the xylem and phloem are typically bundled together in veins called vascular bundles. So guys, remember, vascular bundles are simply veins, and they have both xylem and phloem bundled together inside. And the xylem and phloem that first come and first arise are just the primary xylem and primary phloem. Later on, through lateral meristems and secondary growth, we'll get secondary xylem and secondary phloem, which results in thick stems like woody stems. All right, here are some pictures of everything we just talked about. Let's go ahead and look here on slide 28, and we'll find parenchyma cells, again, very thin-walled, filled with lots of starch, lots of room inside these parenchyma cells. Remember, they have that ability to become totally different cells. Then we have the parenchyma cells. As I said, they have very uneven thickness. Some areas are very thick, some areas are very thin. This is flexible and chewy tissue again. Sclerenchyma, these are sclerids. These are hard, rigid cells. Again, think about that gritty texture there inside of your, that very gritty texture inside of your pear. And then we have the vascular bundles. Of course, this is xylem and phloem all bundled together. Xylem has a thicker diameter. Phloem has a thinner diameter. So guys, this would be phloem here and here. Xylem would be these very large diameter holes, all bundled together, and at the top we even have a little bit of sclerenchyma cells up here that are thick. And sometimes parenchyma. Sometimes you'll see parenchyma down here to support the vascular bundle, to keep the vascular bundle from collapsing and pinching like an artery or vein being pinched. That would not be good, right? Okay, so now let's move on and look at a little bit more at leaf structure and development. So we've looked at stems. We've looked a lot at the different aspects of stems and meristems and primary tissues. Let's now look at leaves a little bit more. So young leaves are produced at the sides of shoot apical meristems in areas called leaf primordia. So leaf primordia are basically baby leaves, and they're produced just to the side of the shoot apical meristem. We'll see a picture of that in just a minute. So on either side of the SAM, you'll find the leaf primordia. Then, once the leaf primordia begin to grow, they'll begin to flatten. They'll expand their surface area because we want more surface area for what? That's right, for photosynthesis, for light collection. Being thin helps also shed excess heat. A lot of plants have thinner leaves just to help get rid of that excess heat. Bilaterally symmetrical is what we typically find in leaves. So if I'm to draw a leaf here, you can see clearly bilateral symmetry, where I can divide the leaf down into equal left and right halves. The upper surface is called the adaxial surface, or the stem-facing side. We find on that side lots of palisade parenchyma with thickly packed cells which absorb sunlight efficiently. So on the upper adaxial or stem-facing side, you'll find lots of parenchyma cells packed very tightly with huge numbers of chloroplasts to absorb the sunlight there. And why would we want to really pack the chloroplasts there? Because that's the sunny side typically, right? And then on the shady side, we have the lower abaxial away from the stem side. We do not have as much parenchyma cells. The parenchyma cells are not as packed. They have more air spaces around them, so they're called spongy parenchyma, and those air spaces, of course, are there for gas exchange. And there are a lot of stomata on the abaxial surface. Now, why would that be, guys? Why would we see more stomata or pores along the bottom side of the leaf? Oh, that's right, because on the shady side, if you have gas exchange happening there, you're also going to be losing water, right? So CO2 comes into those pores, those stomata, oxygen leaves the stomata, and unfortunately, water also leaves the stomata. So we don't want too much water leaving, so we want to put the stomata on the lower surface where it's cooler and there will be less evaporation. Therefore, we lose less water by putting most of that. Plus, it's also good to have the stomata on the lower side because that's where your spongy parenchyma is, where all those air spaces are. We want lots of CO2 coming into those air spaces in a nice, quick, efficient manner. So there are several reasons why that's the case, why those stomata are on the lower surface, on the abaxial surface. All right, guys, here are the leaf primordia. So here's your shoot apical meristem, lots and lots of cell division happening there, lots of undifferentiated cells. Here are your young leaves, here and here on either side. Those are your leaf primordia, guys, here and here. And then we have bilateral symmetry in leaves, as you can see. And then, guys, here you can see even better on the cross-section of the leaf. Here is your palisade parenchyma. Palisade mesophyll is what some people call that. Lots of cells packed in very tightly, filled with lots and lots of chloroplasts. Then we have the xylem and the phloem packed into your vascular bundles, your veins. We also have spongy parenchyma or spongy mesophyll down here with lots of air spaces, not nearly as many chloroplasts down here on the lower abaxial side of the leaf, but we have lots and lots of stomatal pores located here and here at the bottom of the leaf surface. So again, lots of CO2 coming in, hanging around in these spaces to be absorbed for the process of photosynthesis, then lots of oxygen heading out as a waste product. So perfect design to facilitate maximal photosynthetic activity. So leaf veins, again, they're composed of vascular tissue. Xylem conducts water and minerals from the roots on up to the leaves. Phloem carries them on down, and it carries sugars and other products of photosynthesis from the leaves down to the roots. So xylem up, phloem down. I'm sure you'll remember that for the test. All right, guys, let's go on to slide 31 and now look at root structure and development. So we looked at leaves. Now let's switch and now go down to the soil and look at the roots. Dicots, They have a taproot system with a main root that produces lateral or branch roots. Remember, dicots have that thick main root, often used for starch storage and for storage of water as well. Monocots have that thinner fibrous root system with lots and lots of roots, no one main root, and of course they are adventitious, which means they're produced on the surface of stems of monocots and dicots, even in some cases coming right off of leaves. Yes, folks, roots can come right off of leaves, and those would certainly be called adventitious roots. So the monocots produce the adventitious roots in the fibrous root system, but there are some dicots that can also produce adventitious roots. All right, guys, so let's now move on to the summary. So here we have a nice summary of what we have just talked about. So you can see the simple tissues, parenchyma, collenchyma, sclerenchyma, and root endodermis, root pericycle, which we haven't gotten to yet. And these are all considered simple because they're made of only one or two cell types. The complex primary tissues are composed of at least two, but typically three or more cell types, and that would be things like the epidermis of both the root, leaf, and stem, the mesophyll, leaf, stem, or root xylem, leaf, stem, or root phloem. In fact, guys, xylem and phloem produce 4 cells. In the case of xylem, it's tracheids, vessel elements, fibers, and parenchyma cells. Phloem, it's sieve tube elements, companion cells, fibers, and parenchyma cells. The leaf mesophyll, of course, is complex too because you have both spongy, palisade parenchyma, and other cells as well in there. And then secondary growth, which we have not gotten to, this is the thickening process, produces secondary wood, secondary phloem from the vascular cambium and even outer bark, and which is found in the cork cambium. And of course, you can see all the tissues there. So we're mainly focusing on our test currently on primary growth, which is from this point up. We'll get much more into secondary growth as we get into the next test. So guys, let's move on to slide 33 and let's examine youthful stem cells again. So we know the plant meristems include stem cells, undifferentiated cells that have the potential to become specialized and develop into new tissues. Plant stem cells divide to produce one cell when they actually divide. So we're really zooming in now to the apical meristem. We'll find that in the cells of the apical meristems, when one of these undifferentiated cells divides, one cell remains unspecialized, but then another cell is then capable of differentiating into various types of specialized cells. So within the shoot and root apical meristems, you have these undifferentiated cells, and when they divide, one cell remains undifferentiated, so I'll put a little U in there, and the other one has the potential to develop into something specialized. So it either remains undifferentiated or it can become specialized. That's how the cells divide inside of the meristems. Okay? So let's now see what happens after that. So once these cells form and they divide, then it's also not just part of growth. Of course, growth is going to be producing new cells through cell division, as we just described, but let's not forget cell expansion. Cells expand. That's also part of growth. So the main way that plants grow is through cell division, as we just looked at, and cell expansion by water. So absorption of water can really make a plant tissue expand and make a whole plant grow. So extension occurs when water enters the central vacuole by osmosis. The central vacuole, yes, do you remember that from Biology 111? Yeah, the central vacuole is a large container that fills up with water, pushes up against the cell wall because the vacuole gets so big, and that causes the cell to become large and rigid, and then of course results in something of an increased size. So this allows rapid plant growth, and this pressure is made possible. Plant cells under pressure make what are called expansons. Expansons unzip cross-linking cell wall polysaccharides so the cell can stretch. In other words, as plant cells absorb water, the water goes into the central vacuole. The central vacuole expands, pushes up against the cell wall. This causes the plant cell to make expansons, which actually cut the cell wall polysaccharide fibers so it can unzip and then stretch and expand. So let's look and see what that looks like. So going on to slide number 35, you could see here's your vacuole. The water's coming in. The vacuole expands, pushes pressure up against the cell wall. That's called turgor pressure, if you recall. And then that turgor pressure eventually causes expansins, proteins that start to cut the cell wall fibers or unzip them, if you will. It actually cuts the cross links of those cellulose microfibrils, and that unzipping allows the cell wall to expand. Remember, the cell wall is very rigid, but when expansins start cutting those cross links, the cell wall can then expand, and then you have growth and enlargement. So here are here's a more detailed picture of the action of expansins. So before a cell begins to expand, proton pumps increase cell wall acidity. So you've got this turgor pressure building up inside the cell, right? Cells filling up with water. This vacuole is getting really hard and large and pushing up against the cell wall. So then we'll start seeing proton pumps start kicking in. You'll start to see, hydrogen ions pumping out here to the cell wall. And notice you have your cross-linking polysaccharide, right? That's what's sort of gluing your cellulose microfibrils together. So cellulose microfibrils are in green, your cross-linking polysaccharides are in purple. The hydrogen ions start to develop here, and that makes things become acidic. Yes, more hydrogen ions make things acidic, right? So we start getting acidic conditions here in the cell wall. And then the expansin proteins come in with that reduced pH, with that acid environment. You start seeing expansin proteins popping up, and they begin to unzip the cross-linking polysaccharides from the cellulose microfibrils. So that glue, if you will, that sort of zipper of the cross-linking polysaccharide breaks down, and the expansins can then expand, and so eventually they're free to even glide apart, and then the cell can easily expand as the cell wall structure is much less rigid. Isn't that cool, guys? So it all starts out with proton pumps. They pump hydrogen ions out the cell wall. The cell wall becomes more acidic. Then that causes the expansin proteins to begin to show up, and they begin to break down the cross-linking polysaccharide, sort of unzips the cellulose microfibrils. They can expand, loosen up, and then the cell actually gets bigger. Isn't that cool? And we can determine the direction of the growth by how we unzip the polysaccharide. So, for example, here in this particular arrangement, the cytoplasmic microtubules, so the cell's microtubules are arranged in rings, and so the cell wall cellulose microfibrils are also arranged in rings. And so when they are loosened by the action of expansins, the whole cell grows lengthwise. It's kind of like, guys, imagine this. Like say we have a balloon, and we wrap tape around the balloon in a circular manner, and then we blow the balloon up. That's going to cause the balloon to become very long and skinny, right? Tape is going to keep the balloon from expanding into a perfect circle, a sphere. By wrapping the tape around it, it won't be a sphere. It's going to turn into a long, skinny, hot dog-shaped balloon. That's exactly what's happening here. So we could also have our cellulose microfibrils arranged in a different way, and we can get lateral expansion from left to right. So this was vertical expansion because the cellulose microfibrils are arranged in these rings around the outside. Here also rings are arranged on the outside, but they're arranged with their diameter, or I should say, you could say they're, we've kind of done a 90-degree turn here of our cellulose microfibrils. So now we're seeing lateral expansion. So the way your cellulose microfibrils are arranged determines which way your growth occurs. Again, like wrapping tape around the balloon. All right, guys. So this almost done. Just want to talk a little bit about some of these adaptations of stems and leaves. So shoots are formed from modules with 4 parts. We have what's called a stem node where leaves pop out. So check out a stem sometime. You'll notice you have areas where leaves and smaller stems branch out. That's a node. And then those areas between are called internodes. And then, of course, the leaf itself is part of the stem. And then you have axillary merostems, which would generate axillary buds. So what's an auxiliary bud? Well, a terminal bud is at the tip of the shoot. Auxiliary buds are on the sides. So they result in lateral shoots. So in other words, shoots that branch off to the side. So typically we see new branches bear a shoot apical meristem at their tips. So guys, probably a good picture of this will do. Let me see, before I try to draw something, let me just see if we have a nice, here we go. Okay, so this will explain it very well. Let's go ahead and look at that. So we're talking about the terminal bud. So the terminal bud's at the very end. The terminal bud, of course, is just going to make the stem increase in length. Just keep going up this direction. But we have auxiliary buds or lateral buds. And, of course, as they develop, they'll create lateral branches to the side. So if you see a very short, fat plant, like a shrub, that means the auxiliary buds are very active and the terminal buds are not as active. If you see a plant that's very tall and long and skinny, then it has a lot of terminal bud action, but not as many auxiliary buds for some reason. Now, you as a gardener can cut them. You can cut auxiliary buds and you can make your shrubs and trees become more skinny. Or you can cut your terminal buds and make your plants become shorter and fatter. And there's even some hormonal action going on there as well. Notice the node. This is where we have lateral buds or auxiliary buds popping up, also branches. We can also see bud scale scars right here. I don't know if you've ever noticed those scars. Pretty cool looking, kind of bumpy and wrinkly. That shows you where the last year's growth ended. So this particular twig is 1, 2, 3 years old. So just count up the internodes. 1, 2, 3. Each of those terminal bud scale scars represents the end of the previous year's growth. Isn't that cool? So you can you don't have to cut a tree. You can actually just look at the stems to determine the age, at least of that branch, quite well. Okay, and then over here we've got, internodes. Again, areas between nodes. We have the branching of the actual nodes. We have auxiliary buds located here, which result again in lateral branches or branches that make the plant look shorter and fatter. Again, more nodes and internodes, etc. So it's really kind of neat looking. Okay, this is, of course, an herbaceous shoot, non-woody. This is a woody shoot in the wintertime. Okay? All right, guys. Well, we are just about done here. Let's see if we can maybe touch on one last thing, a little bit on hormones. So hormones are going to cause so much of this growth and development. They are behind the scenes. They're causing the cells to become specialized, and they manipulate the destiny of so many cells. So hormones act as signaling molecules that influence development at a site distant from production. So, for example, we have hormones produced in the shoot apical meristem that actually act on areas far away from the shoot apical meristem. One of those hormones is called auxin. It controls production of leaf primordia. It accumulates in particular apical regions, increasing expansion gene expression. In other words, auxins lead to more growth. If auxins cause more expansion proteins, you're going to see more cells expand, you're going to see enlargement, you're going to see a lot of growth there. And they have a big influence, auxins do, on the development of leaf primordia. Auxins also cause apical dominance, which means auxins cause plants to have more dominant apical meristems or buds at the tip wind up becoming more dominant, and so your lateral buds actually get shut down by auxin. So auxin causes plants to become tall and skinny. Gibberellic acid is produced by leaf primordia, not producing NOx. NOx is something that shuts down the production of gibberellic acid, so it slows down the growth there. Because gibberellic acid is responsible for cell division and cell enlargement. So leaves get a lot bigger when you have gibberellic acid hormone around. But if the NOx genes are expressing the NOx product, then you'll see less growth of the leaves. So smaller leaves are oftentimes there because of the active expression of NOx, knocking out that growth there, if you will. Okay, guys, so we'll get much more of this later in class. I want to go ahead and wrap it up and show you just a brief little video of the flowering plant life cycle, which we've talked about so many times before. But let's go ahead and examine that briefly.
The life cycle of a flowering plant is complex and a bit difficult to understand. It begins with making eggs, which, when fertilized, develop into the seeds from which a new plant will grow. Within the plant's ovary are several rows of cells called ovules. These diploid cells undergo meiosis by dividing twice, while duplicating their chromosomes just once. This results in 4 haploid spore cells. However, 3 of them shrivel up, leaving only a single megaspore. The nucleus of the megaspore reproduces 3 times without ever dividing its cytoplasm, and the new nuclei rearrange themselves inside the cell, and cell membranes develop around most of them. 2 nuclei end up in the center, 3 at one end and 3 at the other. Finally, out of the 8 haploid nuclei produced in this process, 1 becomes the female gamete, or egg nucleus. As the flower's ovary is busily producing eggs, its anthers are busy too, making pollen. Like the ovule cells in the ovary, tiny diploid anther cells undergo meiosis to produce 4 haploid spore cells, known as microspores. Next, the nucleus of each microspore divides, but its cytoplasm remains intact. Then, one of the 2 nuclei divides again to form 2 sperm nuclei. The nucleus that didn't divide is now called the tube nucleus. Finally, a hard outer coat forms around the 3 nuclei, and a grain of pollen is created. To make seeds, the male pollen has to get to the female stigma. And since the male and female parts of the flower are separated by a certain amount of space, the process of pollination is usually helped along by insects. In fact, the fantastic variety of colors, shapes, and fragrances that flowers possess show how important it is for plants to attract the attention of pollinators. Once a pollen grain has reached the stigma, the tube nucleus of the pollen grain, followed by 2 sperm nuclei, grows down through the style to the ovary. Fertilization occurs when sperm nucleus number 1 fuses with the egg nucleus to form a diploid fertilized egg that will divide to form an embryo. Sperm nucleus number 2 fuses with the 2 central nuclei of the ovule to form a nucleus that has 3 complete sets of chromosomes. This triploid nucleus will divide over and over again, forming the endosperm that will provide food for the developing plant embryo. The last step in seed production happens when a tough outer coat forms to protect the embryo. Also, in many flowering plants, the walls of the ovary thicken around the seeds to form fruit. In nature, animals that eat fruit help to disperse the seeds far away from the parent plant, so that when the new plants begin to grow, they will not have to compete with their parent for light and water. The last stage of the life cycle of a flowering plant begins when the seed germinates. During germination, the embryo inside the seed wakes up and starts to grow. In the case of most seeds, in this example, a bean seed, germination starts when the seed senses it has enough warmth, daylight, and water to survive. First, a root appears and grows rapidly, searching for water, and seed leaves push up through the soil. The seed leaves hold stored food from the endosperm, but soon true leaves appear and start to capture light energy for photosynthesis. And as this happens, the seed leaves wither away. As time goes by, it starts to flower, making eggs, pollen, and then finally seeds of its own. And when this happens, the life cycle of this flowering plant is complete.
All right, guys, that wraps it up there. It's a nice overview of the flowering plant life cycle. Very complex, but fascinating, which results in pretty much all of our food, or most of our food. Think about all your food pretty much comes from flowering plants, or either directly or indirectly, right? So, guys, we'll wrap it up here. This is online lecture 7B here in your Biology 112 semester, and we'll go ahead and, of course, continue on Chapter 35 in class. Bring your questions there, where I'll see you next.